Let us talk about this fluid thioglycolate medium which is used in the sterility testing. It is a specialized medium which is useful for the cultivation of wide range of bacteria, and this medium is useful basically for the cultivation of anaerobic bacteria, particularly from the blood samples during detection of anaerobic pathogens. And also this medium is very useful for the detection of anaerobic bacteria present in the fermentation product preparation, that is in sterility testing. So this medium, if you see the composition of this medium, it is a nutritionally rich medium which contains all the essential micronutrients, amino acids like uh, cysteine, and micronutrients are provided by yeast extract. Dextrose is the principal carbon source, and the uh, pancreatic casein, which is used as a nitrogenous substance. Then this is one of the principal components present in this medium, that is sodium thioglycolate. It is a reducing agent which maintains a low oxygen tension by removing molecular oxygen from the environment, that is from the medium. So it is responsible for creating the anaerobic conditions by converting it into water. So oxygen is converted to H2O. Another important ingredient of this medium is resazurin, which is an oxidation reduction indicator, that is pink in color. So under oxidized condition it is pink in color. That means whenever it is present in contact with oxygen, the color of this indicator is pink, whereas under reduced condition it is colorless. So it is very important to have this reagent in the medium in order to observe, in order to locate the areas, oxygen containing areas and oxygen devoid areas. Then another important component is agar. So there is a little percentage of agar, that is 0.75 grams. So that makes this medium a little semi-solid. So addition of this agar, it prevents the diffusion of gases, and it prevents the diffusion of gases. So it is responsible for the generation of different zones in a same tube containing medium with different oxygen concentrations, so with different oxygen tension. So in the uppermost part, which is exposed to air, so that part will be aerobic, the middle one will be microaerophilic, and the bottom one will be devoid of oxygen, absence of oxygen. So this is how this agar helps in creating the different zones uh, with different oxygen concentrations. So this medium is basically going to support the growth of aerobes, microaerophilic organisms, facultative bacteria, as well as anaerobic bacteria. After inoculation, you can see this first tube, that is A tube. This A tube, it is inoculated with a facultative bacteria. You can see that the facultative bacteria that can grow under aerobic as well as anaerobic conditions. So you can see growth throughout this tube in the medium. B tube, it is inoculated with gram-positive cocci, which requires low oxygen concentration for growth. So you can see at the top there is no growth, but right from the 3/4 of this tube there is presence of growth. Then strictly aerobic bacteria. So you will find that strictly aerobic bacteria will only grow on the uppermost part of the tube wherever oxygen is present. So at bottom there will be absence of growth. And the fourth tube, that is D tube, it is inoculated with strictly anaerobic bacteria. We can see that the bacteria are growing at the bottom only, and there is no growth at the top. So this is how this medium helps us to understand the oxygen requirements of particular organism. This medium is also useful for the culturing of blood samples wherever anaerobic organisms are required to be isolated. So diagnosis purpose, for the diagnosis of infections by anaerobic bacteria in humans, this medium is used. And another use that we have seen in case of cultivation of anaerobes, detection of anaerobes in order to check the sterility of the particular product.